That's scary. Set my picture of the cell baby.
Are you on Hopkins Wi-Fi or? Maybe just join on your phone. Just like, I just want you to read your question about like an yeah.
I know. You can get out. Oh, your job's so much cuter.
Thanks, Tiff. The brownie. Thanks. Okay. I'll give the intro. Sure. And then also the disclaimer. Okay. Wait, can I use the mic? Oh, just. Hi, everyone. Thanks for coming to our event. My name is Emily. I'm president of the Protein project. But here today is Logan and Shelly from BIOMILQ. BIOMILQ is a women-owned biotech company working on making safe, sustainable, and 100% human milk outside the body, produced from mammary cells for healthier moms and babies. And you guys will obviously learn way more through this slide deck. Logan is a bioprocess development team lead. He's a senior scientist, and Shelly is also a senior scientist. Go ahead. Disclaimer of endorsement: reference to any products or services by BIOMILQ does not constitute or imply its endorsement, sponsorship, recommendation, or favor of the All Protein Project at Johns Hopkins nor Johns Hopkins University. This guest lecture is for educational purposes only, and with a focus on the technology that BIOMILQ has been developing.
Gotta take care of little stuff.
Yeah.
All right. I saw admit, so I clicked admit.
Oh, okay. I was thinking.
Yes. Okay. We're all.
All right, cool. All right, so just like you said, I'm Logan, this is Shelly, and we're here from BIOMILQ, a biotech startup down in the RTP area. It's focused on using biotechnology to and cellular agriculture to produce human milk. So just a quick overview of what we're going to be talking about today. We're going to introduce ourselves in a little bit more detail. Then we're going to talk about cell ag and the overall all protein field. Get into BIOMILQ, our values, our mission, science, and some of the challenges we face. And then at the end, we have time for a Q&A. We don't really have anywhere to be, so we can answer questions for as long as you want. If you have a pressing question that pops up during the talk, feel free to raise a hand. We're pretty low key here. It's not a big deal if you want to ask a question. So just to introduce ourselves, like you said, I'm Logan Howard. I graduated from Hopkins back in 2015. Had a dual degree in biomedical engineering and materials engineering. After I left here, I went to UNC Chapel Hill and I got my PhD in applied physical sciences and material science with a focus in tissue engineering and organ on chip models. After that, I did a postdoc at the UNC School of Medicine with the Marshall Lung Institute. And then finally, I got my first big boy job, and I went to BIOMILQ, and I started as an assay development scientist and have progressed now to our bioprocess development team lead.
And I'm Shelley. I actually attended University of South Carolina for both my bachelor's in public health and I did, bachelor's in public health and master's in molecular cell and developmental biology. My thesis work was in cancer biology, so specifically I studied p53 tumor suppressor. And I then moved to North Carolina, also worked at UNC doing academic research there, and then started at BIOMILQ just over a year ago. And I'm currently a senior scientist on cell line development team. So we focus on acquiring memory cells, characterizing them, and modulating how they produce milk.
Cool. So this is for the Alt Protein Project. So I'm sure we're going to be telling you some stuff that you already know. So I apologize for anybody who already knows that. But here, the best place to start is talking about what is Alt Protein. And in general, it has two different forms. You can have any non-animal derived product that you use for agriculture. That can be plants, fungi, insects. There's a lot of different ways that the field's handling this. Or you can go down the other route of any kind of biotech alternative for ethically sourcing animal-based products. And that's traditionally going to be your lab-grown meat. So two main ways of achieving this at a scale where you can actually have an industry around it is one, you harvest the natural ingredients in a very unique way, where you process them in a unique way that you can get a product. One example of this is that I've seen is genetically modifying insects, grinding them up, and then harvesting their carapaces for like proteins. And the second way you can do it, and the way that we do it, is cellular agriculture. So cellular agriculture on its face is using creating any kind of agricultural product via some new type of biotechnology. That can be tissue engineering, or it can be some molecular biology or synthetic biology. At its core, it really started with GMOs, genetically modified organisms. I'm sure you guys have all seen it, the buzzwords and everything in the news. People scared of GMOs, but that's kind of where cell ag started. Now, it's any company that's using cell culture to guide its growth and to create a product of some sort. That can be food. It can be other agricultural products. There's a company just out of Boston that's using plant cells to grow cotton as cellular agriculture. So it's not just food. It can be other things too. It can also be some things that are a little bit smaller, growth factors, media supplements, basic compounds like singular proteins. And I'm not going to go through this entire history of what this is, but the history of trying to make a meat substitute goes back to the 80s. It started out with, they wanted to make vegetables taste like meat. And it's since progressed, the field has progressed to cell culture based meat. And at least in the last year or so, there's been huge strides in that field in the industry, with the FDA issuing no questions asked letters to a few different companies for cell-based meat. And it's not just history of meat substitutes. There's also the history of using cells to produce something that you can then sell goes back way further than that. It goes back to the 1800s. It's mostly precision fermentation, going way back to you want to make insulin or other enzymes for a product. And just a note here, it says up top, acellular agriculture. We're going to be saying cell ag throughout this, but cellular and acellular agriculture are basically the same thing. Cellular agriculture is just where the cells are the product. Acellular is where something they secrete is the product. So use them fairly interchangeably, so don't get too caught up on the nomenclature there.
So as Logan already said, it's a really vast field. There's a lot of people doing a lot of different things. And so I thought this image here kind of showed the diversity of the field. So, when you think cell ag, you think about the cultured cheeseburger, but there's all sorts of like biomaterials, palm oil, fur, all sorts of things. And where we fit in would be in the dairy sector of this, but it's really expansive and growing every day. And so, not to overwhelm you, but this is kind of what the landscape looks like right now. And the notable thing about this image is that you see the actual companies kind of at the top here, but at the bottom of this picture, you see that there's a lot of support that goes into it. So whether it's material support or technological support and funding. And so it's really a bigger field than just the companies like BIOMILQ that are doing this. And there we are. And so all of the different key players who are doing cell ag, it's consumers, it's governments, it's investors. So here's just some trend lines showing how much the field has grown over the last 5 years in particular. And you can see the slope is just increasing exponentially at this point. So now we're up to about 135 total companies, and you can see that the funding matches pretty consistently. And traditionally, specifically cultivated meat has been the least funded part of cellular agriculture, but 2019 to 2020, there was 6-fold growth in funding. So this kind of remains to be one of the exploding trends in all protein. Specifically Memphis Meats, which did their Series B, brought in almost $200 million.
Get that much.
We'll see. And the US is currently leading the world in a number of companies, but you'll see that Israel and Germany are both really big hubs. UK, Netherlands, Singapore's regulatory landscape has been really, really interesting and is definitely something to watch there. And then I don't know if anybody saw the news that there's a proposed ban on cellular agriculture in Italy that just came out. And so definitely going to be a lot of other countries' regulatory pathways feeding into how this works in the US as well. Definitely a global filters. We went back. There's also really big like meat companies that are investing in cellular agriculture. So I was really shocked to find out how much Tyson and Purdue have been giving to cellular ag companies. And there's kind of maybe a lot of shady reasons for that we'll get into. But the point is the funding has to come from somewhere when it's a startup, and there's a lot of giants moving in. So here's a quote from the Tyson CEO: "If we can grow the meat without the animal, why wouldn't we?" And they're even rebranding themselves now. So instead of being focused on meat, they're saying that they are a protein company. So they are kind of leveraging this potential to get involved with cellular agriculture from the jump. Whether that's good or bad remains to be seen. And so the other important factor of who's doing cell ag is definitely the consumer. So this graph up here in the top is showing the plant-based industry. It's increasing rapidly for a variety of reasons. So a survey of consumers in the US and Canada, the main reason to do it has been to improve overall health. But there are reasons of allergies in traditional products. There's concerns about the environment. There's a desire to just eat clean, whatever that means to the individual person. And this trend is mostly being driven by millennials. They're a really large market sector with a lot of money to spend. And so they're the ones spending it. So a survey said recently, I think I read 30% of millennials will eat a plant alternative at least once a week, and 30% daily, 50% once a week. The interesting thing to look at to me is this was a survey that was done in the US and the UK about how open people are to trying cultivated meat. And it looks like Gen Z is going to be even higher than millennials. So the trend is only going to grow. The industry is only going to get more and more popular. And even my boomer dad said he would be willing to try it. So it's not just the young people, but they are certainly driving it. The other major key player is governments. So how are they going to regulate it is one question, but also government funding. So the Netherlands, there was just a huge amount of money. I think it's 60 million euros dropped on cellular agriculture in the Netherlands. And you guys probably heard about the USDA giving, I think it was $10 million to Tufts, which is a huge research hub for cellular agriculture. And you can now get a degree in cell ag there. Johns Hopkins. A lot of chapters too for cell ag. So I think it would be better to hear from you guys why you care about cellular agriculture. Since you're all here, I'm assuming that you all care. So why do you care? Yeah.
Like I don't eat fly fish, but I was treated. And I did that in the environment. And then animal welfare too. Care about, but like it's definitely a big environmental faculty, and then I think that kind of relates into sort of like because I know
For sure
that like the environment, state of the environment is more affecting people who are contributing less to the negative like to hygiene.
Exactly, yeah.
Absolutely.
There's that disparity there. for sure.
Close.
Yeah,
Yeah, like, the need for a beef alternative is not just like farming and it's really depressing because we do something to go increasing, so it's kind of just like, do something else. Need another option.
yeah. And the population is expected to, what, double by 2050? We don't have the space.
25% of all the land, like all the plant surface is used for agriculture, specifically for livestock agriculture. It's a fun fact. Anyone else? Why are you here? Why do you care?
I have my reason for BIOMILQ specifically. I have an 8-month-old son at home, and it just is both a blessing and also a pain to breastfeed. When he was 3 months old, I had a terrible problem with appendicitis.
Yeah.
That lasted 2 months, and it was just so painful, and I could not do any work or continue to go through my life. And at the same time, I feel guilty that I was not able to do it because clearly the benefits of breast milk is just so much better than formula. And I feel like the technology that BIOMILQ is trying to bring to the market is just amazing, and what the world needs. And also, I'm a PhD student at Hopkins. I work on a lot of clinical experiments, so I want to one day to file them.
Awesome.
And thank you so much for sharing that too. Like that is, I mean, to hear from people who are struggling and who actually benefit from it, it's like a lot. And thank you.
That's why we do this job.
Exactly.
That's why we picked this company. So, anyone else feel like?
Yeah.
Yeah, for me, it's just like small scale industry, like industrial waste. There's so many. It's just
Right. Yeah, I think these are all fantastic reasons. I think you guys hit pretty much
Most of the things that we
everything.
would have talked
Yeah.
about.
Economics. One thing, this is a little bit dicey because it goes both ways. If you use cellular agriculture to reduce jobs in conventional agriculture, I think we were telling some of you we're from rural South Carolina. None of us take job loss lightly. So there's work that needs to be done to ensure job security as well. But one economic benefit is that there are a lot of surpluses and subsidies that come along related to conventional agriculture that then get passed on to the taxpayer. And this feeds into social equity as well. Like you pointed out, you said it very well. It's the ones creating at the global level and at the city level. The ones that are creating the lion's share of the product are not the ones that are necessarily being fairly treated for that. So, yeah.
On. So we've painted like a nice picture of cell ag and the things that we want to do with it, but it's not sunshine and roses. It's not just sunshine and roses. It has a lot of limitations and roadblocks too. So we're going to open it back up again and so what are some problems? You guys are aspiring scientists. You are scientists. You probably have some ideas about what you think the major challenges are going to be. I'd be really interested to hear what you think those are.
Absolutely.
100%.
Have so much power globally, and trying to overcome basically huge loss of overcome funding, governments lobbying, and so even these politicians and stuff are making the laws and putting tax regulations are also the event.
Yeah, absolutely.
We're going to talk a little bit about that.
Regulatory for huge troubles too. Yeah. Very hard.
Yes, it is.
Yeah. A million %. Yeah. I think for us, it's been challenging to define what our product will never be in a million years. And we've had to like, as much as we want it to be like the absolute perfectly identical, you bring up a really good point that there's limitations there. Oh, yeah.
I think also like just because the products is very, very the initial costs, they're initially kind of very expensive, and they're not very accessible to certain groups of people, which makes it harder for some people to want to necessarily go that direction.
Exactly, yeah. And I know, it's hard to think about that in the short term. You know, the last thing you want is to create, I think our founders said, like another elite product for another elite child. We don't want to be making social inequities worse. That's certainly a huge consideration, absolutely.
Yeah, I think you guys hit most of the
Yeah.
important points that we were going to talk about.
Yeah.
But I'm going to go into a few of the different things that some of the critics say, and they may very well be right. Production costs are too high for a widely accessible product. This is Dr. Mark Wolf, I think, Mark Post at Maastricht University. This was that initial hamburger that was made back in 2013. That cost the project that led to that cost $330,000. $330,000 for a petri dish that's the size of a Big Mac patty. That's what came out. And this right here, this is a bunch of stack flasks where the cells for that patty were grown, and this isn't even all of them. It just in terms of volume, space, media consumption, all that, just for that. So one big criticism is, yeah, production costs right now are too high for a widely accessible product, and that's where a lot of the biotech sphere is going. We need to bring down costs if this is going to be a viable industry. Second thing, we're making a lot of estimations here based on assumptions. So our estimated environmental impacts for energy use, water use, land use, they could be overly optimistic. In fact, shown here, this is a study 3 years apart, where the only thing that was changed in the projections was bioreactor sterilization, sterilizing your product so that you don't have bacteria in it for scale up. And you can see the energy use from that increased an extreme amount, as did the water use, just for sterilization, and that's just one estimation. So it's entirely possible that these projection systems that we're using are too optimistic. Something that we were talking about earlier is the funding structures that'll actually pay for biotech startups like Tyson. The money has to come from somewhere, right? So if those giants get in and they buy up all these cell ag startups, it could very well lead to more power asymmetry in the food system. The people who actually control who's getting the food, who's making the food, could be in the hands of those wealthy elites, and that could be a major problem. And then here's just like we had those 3 slides, but there's a list here of so many different things that the critics can say. Taking attention to resources from other solutions that may be more practical, possible. That the technology is dubious. I disagree with that a little bit more, but pad protection reduces the necessary transparency. That's something that we work on, contributing to literature while also having a proprietary technology. It's kind of difficult. Market will remain niche. You're talking about your family is completely not open to it, and there are many, many families that are like that. Won't do for the inequities in the global south. That's pretty accurate. It's too susceptible to disruption. That's kind of a harder one, and it comes down to a lot of funding. Less ethical, it's still less ethical than plant-based alternatives, and it fetishizes meat. I find that particular quote a little funny, but it's a valid point. Like you still have to have the animal source come from somewhere if you're doing something like this. It's going to create a meat-free elite that shames the working class. If it's an expensive product, you're not going to have access to it as a lower class, and we don't want to do that. Not enough fundamental questions have been answered, and this is one I agree with as someone who works on this daily. We're still very early on for this to be a primary industry in the world. So we need to answer a lot of more fundamental questions, both biologically and for expansion, for that to be true. Consumer acceptance is overestimated, again, coming back to people not accepting things that they view as unnatural or things like that. It comes close to the real deal, but we lack the technology to make 100%. That's going to probably remain true forever. That's a valid criticism, but we're trying to get as best as we can to make the world a better place. And then finally, the corporations' policies will shut it down. You mentioned big agriculture companies and things like that. They have a lot of lobbying power, so that's entirely possible. So that's a lot of reasons to throw at you for why cell ag is not great, but it doesn't overshadow the fact that we should still pursue it as scientists and as citizens of a global economy where we're trying to make the world a better place.
Yeah, so I'm going to step in and talk a little bit about Enter BIOMILQ, what we stand for, our mission, our beliefs, our founding, and then like how we're making it happen. And feel free to stop me if you have specific questions, but we'll certainly open it up at the end for questions as well. So to give you a little bit of background here, 2021, there were over 3 million babies born in the United States, and that number is increasing. We have pandemic babies. This number is probably going to go up over the next few years. And the reality is that there's pretty much 4 options to feed your new baby when you have a new baby. I will point out that when I go to the gas pump to refuel my car, I often have more options than a new parent who's trying to literally feed their baby. So breastfeeding is absolutely World Health Organization recommended, CDC recommended, best way to feed your baby. They recommend breastfeeding exclusively for an infant's first 6 months and then beginning to integrate foods gradually, but breastfeeding for up to several years. The reality is that 84% of parents in the United States do not breastfeed for 6 months. So there's certainly a gap in what's recommended and what actually ends up happening. So that brings us to infant formula. This is a really safe, convenient alternative to breastfeeding. It is sourced from bovine milk, so cow milk and not human milk. 90s baby here, infant formula all the way. So things have certainly changed a lot in the last couple of decades. And then milk banking and milk sharing, these are kind of similar. They both involve getting donor milk from another human. Milk banking is a lot more formal, much more accredited. It's typically for NICU babies specifically, but there's a lot of regulations for sterility, for content, these kinds of things. Whereas milk sharing is much less formal. It could be peer-to-peer. Someone down your street can give you breast milk and there's no regulation for it. And this is not to say that all milk sharing is bad, but there is a risk of contamination. There's a risk that it's not what the donor says it is. There could be, cow milk thrown in there. So it's not, necessarily the best option. So breastfeeding is definitely by far the best gold standard. There's many, many benefits to your baby. So there's benefits for gut development, immune development, neurological development. It's related to lower rates of obesity later in life. It's beneficial for the mom too. There's lower incidence with breast cancer and with cervical cancers. And there's emotional benefits too. So a mother bonding with her baby, there's hormonal signaling that's going on for both parties that is really beneficial. But as I said, the reality is that most women, it's actually lower in America, so 38% around the world, and I think 84% in America do not make it that full 6 months. And so we do want to be really careful with the language that we use. There is, breast is best, absolutely, but at the end of the day, fed is best, and we want to be really careful about that, where we're not shaming moms who cannot breastfeed, who do not want to breastfeed. And so I put this little cartoon on here, the fact that, anybody who's saying, breast is best is maybe not considering the reality that we live in, where we don't have the support that we need for moms to breastfeed. So I will, again, open it up to you guys. Breastfeeding is difficult or impossible for many reasons. Can you guys maybe think of some physical reasons? I know we've already heard one. So I'll open it back up to you. Physical reasons why it may be difficult or impossible. Yeah.
I know that's something preventative instead of getting good.
Correct. Yeah. 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 You physically can't. Yeah. Breastfeeding can take up to 4 hours a day. So if you have nowhere to do it, yeah, all the time. All the time. Yeah. Other reasons. Exactly. Yeah, we have adoptive parents, foster parents, parents in the LGBTQ community, and also parents who maybe have a communicable disease and they don't want to pass that on to their baby. Other reasons? Yeah.
Somebody in the chat said children are on that.
Yes, so sometimes the baby is unable to latch. So this can be a difficult problem to rectify with an infant where they cannot physically latch onto the breast.
Physically, one of the big ones is sometimes, mothers like humans are very different. Sometimes mothers just have a low milk supply for whatever reason, genetics, upgrowth, anything like that.
Medications,
They
diet.
have a low milk supply. That's another physical reason why it sometimes doesn't happen.
So there's emotional reasons as well. So any guesses about emotional reasons? Yeah. Absolutely, and there's actually studies showing correlation between postpartum depression and lower rates of breastfeeding. That's a very real thing. Yeah. That's a lot. Yeah. Right, and your body responds to that stress. The more stressed out you get, the more cortisol's up, the lower your milk supply gets. So it turns into a feedback. Yeah. Very much, yeah. Yeah, I'm sure you can. Yeah. Yeah, and I don't know, I mean, you guys probably understand a lot what it's like to be sleep deprived. Said you pulled an all-nighter last night, right? Not a good feeling, not great for your mental well-being.
And those kind of lead us really greatly into the next one.
Yeah.
Societal reasons.
So you've already mentioned lack of paid maternity leave, paternity leave, so you don't have necessarily somebody supporting you and helping you. Workplace, yeah. Yeah. We saw one on the lunch flight.
Absolutely.
Yeah, and then people are just like staring at you, like judging you for doing something that you're supposed to be doing. Yeah.
I was kind of related to that, I felt like there's non-private areas to one person would let say you personally don't hear about being like private breastfeeding people, you scared people are gonna be upset.
Yeah.
I don't know if that's happening. So this comes from what I was feeding about private and then public.
Right.
That's a full.
Yeah. And then it's like, well, why are you feeding your baby formula? Yeah.
Again, growing up in rural South Carolina, I saw that growing up, like people will walk up to a breastfeeding mother in public and be like, why are you doing that in the restaurant?
Right.
And it's a completely
Don't
natural
wanna
thing.
see that, yeah.
So
Absolutely. That, I'm sure that takes an emotional toll as well.
any other societal reasons you guys can think of?
Yeah. It's depressing, but I think you guys hit them all.
So that all kind of leads into why we're a thing.
So, as somebody who has never lactated, I think it's more helpful, like, to hear your testimony, but also I can show you guys what other moms have said. And these are more meaningful. So there's a lot of reasons, like we just listed out. But the bottom line is that parents need more choices. If you want to breastfeed and you can breastfeed, that's fantastic. You should definitely do that, but it's just not necessarily, it's not as easy as it sounds like. And so there's a lot of limitations to infant formula. You guys, can you think of any like nutritional? Yeah.
Nutritional, might not be nutritional, like, but my mind is like the rising product of,
It's insane, yeah. The little tin. Yeah.
So like, obviously, you get the bad.
Yeah.
So that's something that.
Your baby is missing. Yeah. Exactly. Like a recombinant, like to spike it in or something. Yeah, I do think that some formula companies are doing that. I don't know off the top of my head.
I know Danone in particular is.
Okay. Yeah. But at the end of the day, you know, you have to be very cognizant of immune responses to that.
And these are mostly meant as guides. If you have any reasons where you think,
Yeah.
like things you might see limitations.
Yeah. Emotional, environmental, yeah. Lactose, yeah. You have to get a special lactose-free formula, which in the infant formula shortage, moms are taking anything that they could. Yeah. Yeah.
Like letting her have less of that bonding, which I think is really important for their cognitive development.
Exactly, yeah. Any other reasons? Environmental toll of the dairy industry is quite high.
30% of all of our air, all of our fresh water goes to livestock agriculture. 15% of our greenhouse gases come from livestock agriculture and from the bovine industry. So.
And deforestation to make space as the population grows. Yeah.
So those are a lot of reasons.
Yeah. So BIOMILQ, I did a quick search right before I came, and the bottom line is that there's just like not enough attention being given to how we feed babies. So if you look up erectile dysfunction, 6,536 results, low milk supply, 819. It's shameful. Yeah. And parents ultimately just need more options. Like I said, I can go fuel my car and I have 5 buttons that I can press. If I'm a new parent, I have 4 choices, and one of them may not really be a choice. Multiple of them really may not be a choice. So I'm sure you guys probably heard about the infant formula safety recall, still going on, but it was, you know, at its peak about a year ago. So this is just showing, you know, parents really need another choice, especially when something like this happens with supply chains. It just really, really drives that point home. And so that's kind of the environment in which we emerged. So BIOMILQ was actually officially founded January 2020. Our whole mission is about empowering parents to make these choices. So we will never officially be, you know, breast milk. As you pointed out, there's, you know, components that you just cannot get. So we are cell cultured human milk, and it's the goal is to get as close as possible to the nutrition of breast milk with the convenience of formula. And we also take pride in being, you know, women-owned, science-led, mother-centered. We all take that very, very seriously where we work. So it's all about a choice, just having one more thing on the table. And this is our co-founder and interim CSO, Layla, with her new baby, Violet. And Layla actually was cell biologist by training, and she struggled to produce enough milk. She kind of got this, what everybody else said was, you know, crazy idea, watching the Mark Post burger, and said, why can't we make mammary cells do this? Secrete milk products. But I think she's also a very eloquent person. She is very good with words. So I like this quote by her. I'm here to invite moms who have already endured so much shame for feeding formula to consider what they would choose if they truly had a choice. And to me, that really resonates. Again, all about the choice. So it's agreed upon by the NIH that we don't know really anything about milk. We don't know what it is. We don't know a lot about how the body makes it. We don't know a lot about how it actually benefits the baby. So how we're making this happen is we are learning a lot of the basics. So we are going in and really reinventing the wheel. And so this is kind of our overview of what this looks like, specifically from what we've called our breast milk study. So we have donors donate milk to us, and we take cells from that milk. We also take the milk and analyze it to get an idea of what milk actually is. And this pretty much involves driving around Raleigh-Durham area with a cooler and knocking on someone's door and saying, "Hi, I'm Shelley from BIOMILQ, and I'm here for your breast milk," for people's offices, you know, wherever they happen to be. Once that milk is received in the lab, the cells get separated out, and then we grow them up, and they start at a very low density, and they just keep dividing, keep dividing, keep dividing. Once we have enough cells, you saw the picture of those stack flasks, those get seeded into a bioreactor. And that's when the cell surface goes from being grown on plastic, like a Petri dish, to getting a 3D shape. So Logan will talk more about it, but we brought a prop. So that's a bioreactor, just a 3D structure. And then the cells are induced with different hormones to produce milk, and they synthesize the milk, and they will secrete it. And then we anticipate lengthy safety regulatory checklists that we can go through to make sure our product is safe before it ever appears on the shelves. Any questions about like the general process? Yeah.
So, when you said I don't know examples from people, how many people are we talking about? Like, what's the
So I think the last I heard, we had over 100 donated cells, I believe.
100 lines, yeah.
Yeah, and I think even more donated for compositional analysis. And we worked really hard to make sure that it was as inclusive as it could possibly be. That's something that we all care a lot about. So as we're, you know, doing this research into actually like what is milk, that we're getting a reflective sample. Hey, Renee.
Hey.
And so a lot of this comes from really basic mammary cell biology. Like I said, there's not a lot that is known. So we take basically, I'll run you through it. So this is a human breast that has changed during the 3rd trimester of pregnancy. And you have these terminal ductal lobular units, which are the sacs basically that create milk. And this is pushed through a duct and given to the baby through the nipple. And then you see here that it's lined with cells. I don't know if you can actually see. Oh yeah, you can. Okay, so it's lined with cells along the duct and also along these lobular units or buds. And there's multiple types of cells that actually make up these sacs. So they're surrounding this hollow lumen here. It's kind of like a balloon, but these are called luminal cells. And then you have myoepithelial cells that surround them. Luminal cells are the ones that actually synthesize milk. They're surrounded by these myoepithelial cells that provide the contraction to push the milk out into the hollow lumen, where it then goes down the duct to the baby. And one thing I want to point out is that there's also like supporting cells here. So you have fat cells, adipocytes, and connective tissue fibroblasts. So we're having to learn a lot about this to even understand how the milk is getting synthesized. Yeah. So there's definitely discussions going on about what method is best right now, for sure. We don't know the answer to that question. Yeah. So are you talking about in the body or the way that? Yeah, Logan will talk more about that. We have a specific.
But in general, we have a cell processing. One of our colleagues, Victoria, was kind of the head of cell processing, where we would get the milk sample, spin out the cells. There's a lot of cells in breast milk.
Yeah, you'd be surprised. Cool. So as you pointed out, milk changes a lot, not just over the course of lactation as, you know, a 6-month to 2 years, 3 years process, but even in one feed, you'll have foremilk and hindmilks that are different compositions. There's different fat content. So foremilk is a lot more watery to really hydrate the baby. And then hindmilk has a lot more fat content in it. That's a lot of the caloric volume. So we're having to figure out what even is milk, how is it changing over time? And then there's also, I've put here the nutritional profiles for colostrum, which is really premature milk that's only the first few days after birth, versus mature milk, and then versus bovine formula. And what you see is that they're different not only in the macronutrient profiles, but also in the caloric content. And this colostrum, you can see the volume of milk is changing as the baby's stomach grows and develops. And I think you pointed out as well, there's a lot of bioactive components in breast milk. So it's not just like you have, you know, your protein and you have your lactose and you have, you know, your lipids. There's cells from the mother, which is how we get our input. And then you have, I think, 800 strains of bacteria, which our product will never be able to have because that goes against aseptic technique. But there's also all sorts of proteins that do specific enzymatic activities related to immune modulation, gut development, and acting as helpers for other nutrients. So these are things also that we have to figure out and consider what our final product is going to have. And then another thing that we have to figure out is how are we even going to drive this lactation? So lactation is normally driven by hormones. So you can see here the two main ones being oxytocin and prolactin. So this graph is showing hormone levels at pregnancy, birth, and then postpartum. And what you'll see here is throughout the 3rd trimester, prolactin expression increases a lot. And at the onset of labor, you get a lot of oxytocin. That's what leads to uterine contractions. So tons of oxytocin. And that's also what drives that myoepithelial layer to contract. However, prolactin is the hormone that causes the actual synthesis of milk. So you need both of them. And you can see that they happen in waves here. So if you see this black peak, you can think of that as like a release versus the green peak here is like actually make the milk. So you have waves of hormones coming in, perfectly coordinated in tandem. And what's further complicating this issue is that a suckling baby is what would normally lead to these hormones to be secreted from the hypothalamus in the brain. So how do we replicate that in cell culture? Cannot have a baby. That's not something we can do. So definitely a lot that we are still figuring out, a lot that we still will need to take into consideration. And one other thing that we are doing as a team is, Logan pointed out, trying to contribute to the literature so that we're actually helping the problem as we go along. So these are 2 team members, Claire and Zach, who recently presented at the International Milk Genetics Consortium. So they did a poster on the metabolomics of milk. And so they're taking these samples that we got in our breast milk study and analyzed to actually contribute to the literature. I'll hand it over to Logan now to tell you about bioreactors.
So as she was talking about, this is a very complex bioprocess. So that makes my job kind of hard. So the big main point here that I'm going to be talking about is bioreactors, and specifically hollow fiber bioreactors. That's just kind of this picture here. There are many kinds of bioreactors that I'm going to talk about, but this is the one that we're currently focusing on. And we brought a little prop, like I said. So what is a hollow fiber bioreactor? So this right here contains 4,000 square centimeters of surface area for cells to grow on. Basically what this is, you have a cartridge, a number of hollow fibers all the way running through it that are semi-permeable. And the way we do it is, one, you have cell culture media, a big reservoir tank that goes through and you push it inside of the fibers. But on the outside, to these little outlets right here, you can seed your cells. You give them time to grow to confluence along your fibers. And what happens is they grow to confluence along the fibers, basically creating a barrier between where your media is going through the fibers. As you can see right here, you have your epithelial layer of these cells, and then anything they secrete coming out is going to come out and not be contaminated by your cell culture media. There's a lot of scientific, there's a lot of building up to that to make sure that the cells are actually forming a non-permeable membrane, that they're secreting things that you want, but this is the general goal that we're going for with hollow fiber bioreactors.
How big around is it?
So this is, like I said, this is 4,000 square centimeters. Yeah, this is 4,000 square centimeters right here. So almost half a meter of cell culture space in this right here. We have one that goes up to about 5 square meters. And in the literature and what you can buy right now is up to 10 square meters of cell culture space just for one pathway. That's a lot of cells. That's billions upon trillions of cells. So, yeah, I'll probably hand this out if they want to.
Yeah, do you want us? Yeah.
Yeah, we can hand out a prop.
Be careful. But,
Yeah. Yeah, it's just a bundle of fibers going through. So just to show that we actually can do this, we brought a pretty picture. The blue cells are just a nuclear stain showing that an epithelial cell layer is there. And the red, which is a little bit harder to see with the lighting, but that's actin, showing the cells aren't just there, they're actually forming some kind of microstructure. So some of the pros of this, like I mentioned, is separation of product and media. If we're trying to feed babies, we don't want to be feeding them DMEM. We would like to be feeding them a human milk product. One thing with this, hollow fiber reactors traditionally have a very low stress rate, which puts low shear on the cells, which can lead to, which is good so that it doesn't actually kill them. And then one big factor in tissue engineering is that it's not just a 2D monolayer that's good. 3D growth is traditionally going to be needed for any kind of in vivo like behavior. So hollow fibers allow for that to happen. They allow for 3D growth at very high densities. But there are some cons to hollow fibers. It's basically impossible to get an accurate count of how many cells you have in there. Getting them off the fibers, counting them without shearing them, blowing them up by accident, it's very difficult. And then you come into transport phenomena and fluid flow. Actually getting uniform seeding across the cartridge so that they're growing uniformly across that entire 4,000 square centimeters is extremely difficult. And then the next two kind of come together. The architecture and the microstructure there aren't like the body. It's not biomimetic. And replicating all that mechanotransduction factors, replicating the stiffness, replicating the porosity, replicating all those mechanical factors that you would find in vivo is extremely difficult. And then finally, scale up isn't a linear process. When you go from 4,000 square centimeters to 10 square meters, you're talking billions and trillions of cells. And as you get to those higher densities, they act differently. So we're using hollow fibers right now, but there are a number of different ways that the cell ag industry handles this. One, and the one that you most likely heard of, if you've heard of them before, is stir tank reactors and suspension culture. This is kind of the gold standard in the biotech industry and the cell ag industry. It's very well developed. It's been being developed for years. So basically what it is you have this big suspension stir tank. You have an impeller that goes around and around in the middle, and you have suspension cells that are sitting in there and producing whatever product you want. Has really good mass transfer. It's really easy to pick out all of the secreted products that you're getting. Keeps a good mixing of the cells. What can happen in adherent culture is you get some certain subpopulation that's doing one thing, and then this other population over here decides they don't want to do that thing, so they're making something else, and that's not super efficient for a scalable industry level model. And then there's a high potential for volume-based scaled up. I talked about the biggest one you can buy for a hollow fiber is 10 square meters. You can get up to 1,000 or even 10,000 liter tanks for stir tanks, depending on what company you go through. And again, for sampling and monitoring purposes, doing this in suspension is way easier than doing it for adherent cells. For adherent cells, you have to somehow pull them off the membrane to analyze what they're doing. And with suspension culture, you just pull out a mill and you can go test it to your heart's content. But it does have some cons too. The big thing, especially for us as a product related model, is there's no separation of media and product. It's all your media is in here, all your secreted products in there. It requires immune precipitation and other different ways of isolating out your product. It can be really difficult when you get to these big sizes to get adequate oxygenation and other gases to get to all sides of the stir tank. So again, that leads to some subpopulations, which isn't super efficient. Like Like I said, this impeller is directly in contact with the fluid that's hitting your cells, which can lead to high shear forces and can kill them. And when you're in this volume, like once suspension cells hit a certain volume, they tend to not like to grow anymore. So there's an effective limit on how many cells you can get per mil of suspension culture. And finally, and this is probably, I have a material science background, the one that irks me the most, is you don't have a mechanical or a substrate reaction with the cells and liquid. There's no ECM there. There's no basement membrane. Yeah, there's nothing like that. So you're not creating an in vivo like environment here. Even if it's scalable, it's not super like the body. So that's stir tank versus hollow fiber. I could spend 4 hours here talking to you about different kinds of bioreactors. Here's just a list of, typically they're going to be either immobilized or suspension based. There are dozens of different iterations of what those look like, and it's going to change based off what your needs are. As we develop our technology, our reactor's design is going to change based off what we're doing. And it may very well become its own version. Just so if you're ever if you have any questions about that, we can leave them for the end, and I can talk about some more styles. So also talking about, limitations of what our technology looks like. I talked about what we're using in hollow fibers, but there's more than just the specific bioreactor and that we're trying to make. If you guys remember that little meat patty that cost $330,000, scale up of cell culture is extremely difficult once you start getting to these very, very high volumes. A number of different challenges come here. It can be media costs. Media is extremely expensive. Once you start getting to these really high volumes, we run through liters upon liters every month. Any costs associated if you're doing adherent culture, these extra metabolites or stuff that you might use to put on the surface to make it more biocompatible, those get extremely expensive very quickly. Just having like that space. You remember you saw this stack flasks where these cells were grown. Just that volume of space can be extremely expensive. You need incubators, you need rooms, you need people to monitor them, you need gas flow. It's exponentially more expensive the bigger you get. And then kind of the big one is long time cell culture. Long time scale cell culture. Cells aren't designed to live forever for the most part, especially not ones that you would eat or that might produce breast milk. They're not designed to live forever. So keeping them going for long enough to make a lot of products can be challenging. Media development, I mentioned it's extremely expensive, but we have an entire team dedicated to media development. Does it contain all the growth factors that your system needs to actually produce the product that you want? That's an extremely complicated cell biology question. You have to test a lot of different iterations. Does it give you the phenotype that you want? It's not just things that they're making, it's how do they look? Are they epithelial? Are they myoepithelial? Are they mesenchymal? Did they go back to a stem state? It's extremely complicated when you're looking at phenotype to make sure that the media is accomplishing what you need. And then some of the big questions: how do you make it cheaper? How do you make it animal free? And how do you make it safe? Again, we're trying to make a product for babies, right? You want to make sure that it's safe. If our media isn't animal free, then that's not very good of us for a cell ag purpose. And then again, all these supplements, all these hormones get very expensive very quickly. So you have to be able to make them cheaper.
Yeah. And then from the cell biology perspective, I might kind of took you through it already, but basically it's a moving target. So can we take these cells and put them in an environment that stresses them out and try to make media cheaper at the same time, while also trying to get them to grow more? But then also they have to, mimic a very complex organ doing a very complex thing. And so there's a trade-off. So you always have to consider, what is the cell capable of? How much can I get away with in terms of like reducing and simplifying? And so this is just, I get like really into all of the cells, but this is one of my favorite populations of them. You can see there's like a very heterogeneous appearance, how they all look. So really defining what the cells are, how do we characterize them, and what are they going to produce? It's incredibly complex.
And then one last thing is regulatory. It got mentioned earlier, but the regulatory pathway for cell ag in general and for us specifically is extremely complicated. When you're talking about feeding something new to people, the FDA has something to say about it. You have to ensure that all your cell lines aren't pathogenic, that that they're not infected with anything from cell population. You have to ensure that they have the chromosomes and the karyotype that you think they do. These cells are what you think they are, genetically. And then figuring out if something that is safe to eat or if it's going to be toxic at higher levels, that's going to require a lot of different testing down the line for a regulatory pathway. And then talking more about cell ag in general, this is a very new field. And the FDA, it's getting better, but defining what is safe, what's allowed, what isn't allowed is still very, very much in its infancy. And that's something that we're dealing with even more because specifically for us, our product is aimed at the most vulnerable population, subportion of our population, which is infants. So if we're selling something, we have to make damn sure that it's healthy, it's safe, and it's not going to hurt anyone. So that kind of leads us to the end of the talk. What's next for us specifically? We got our Series A, like 18 months ago? Something like that. We got our Series A funding of $21 million about 18 months ago. Right now, we're in between Series A. We're getting ready to go for our Series B fundraising round, which will be ideally for more than $21 million. And then if you want to talk about the.
Yeah, so I think that 2 recent no question letters from the FDA are really paving the way. So Good Meats is one of them and Upside Foods is one of them. So this is not, the FDA saying like, oh, everything's perfect, but it's saying no further questions so that they can move on to the next part of getting this product to market. And so we're really encouraged by that. I think that's a sign, like Logan said, the FDA is getting a lot more communicative. Instead of saying, well, you decide how you want to regulate it. They're giving us actual feedback. It's turned more into a dialogue. And so this is all really great news moving forward. So we're very excited.
So I think that takes us to the Q&A. So you guys feel free to ask us any questions about BIOMILQ, cell ag, life, industry, career path, stuff like that. But we also have, in case you guys are interested, if you want to follow the company on any of these QR codes, and we have business cards for both of ourselves. If you want to take one of those for each, where you can reach out to us. So I'll just open up for any questions you might have.
Yeah. Very good question. So it's going to be probably several years from now, whatever we are putting out in the short term is not going to be what it is in the long term. As we're fine-tuning this technology, it's going to change. As we're getting the behavior of the cells to be more like a lactating breast and less like cells from in a bioreactor, it's going to get more and more complex. So there'll definitely be like probably some kind of intermediate.
Are you planning to use genetically modified cells or just use the cells from, I guess naturally, I know that might cause some more FDA regulation and also for meat.
Yeah, it's definitely an open question. There's pros and cons to both. I love to like nerd out about this kind of thing. I'm limited as to what I'm actually allowed to say, but it's a very good question. And nothing's off the table.
I know you're out there really anti-chemo, but here in the US, we're very
Yeah. Yeah, from what I can tell you, website just released their like FDA as well, and so there's some good information there that we're looking to see kind of like what kind of modifications did they use. Our final product is going to be, like ideally not like everything is absolutely, to a T perfect. But yeah, definitely open for now. Yeah.
and then are you planning
A big part of that is making it accessible to everyone. It comes down to on the technology side, making sure that our media, like we can make that in-house as cheaply as possible to still get a viable product. That's actually one of our biggest questions. Like like she said earlier, we don't want to be one of those companies out there for the elites. That's not why we're with the company. It's not what we're trying to do. So approaching it from a bottom-up approach to make sure our process is as cost-effective as possible will ensure that our future product is as cost-effective as possible. That's the approach that we're taking. What was your second question?
Yeah, I think the big thing there again is breastfeeding is absolutely gold standard, and just really trying to give as much transparency and trust as possible, I would say. We're both scientists, we don't do a lot of the marketing, but, I think all of us feel very, very strongly we would not be there if it was going to be, something that was like predatory in any way. They want to, instead of taking advantage of these populations, make everything absolutely above board, and our product's not for everybody, that's the thing, celebrating. We all want the same thing at the end, it's just healthier babies, healthier moms.
I think Layla said it best. It's a choice. We want to give just another choice. That's all we want to be.
I think. So, I guess kind of because it's like how you really cells, using, like, I guess they have to tell people to use their cells, especially like, when you're talking, like, with some people, so.
Yeah.
Thinking about like, like a comes back.
Yeah, so we have IRB approval, everything is entirely voluntary, you can opt into, the study, and you can opt into donating cells as well. So this is through our website, and there was lots of, informed consent, somebody going through everything. So that's pretty much just standardly going with all of the regulations that are out there. One other way that we do get our material that hasn't been as successful and isn't necessarily what we'll be doing moving forward is from elective breast reductions as well, and that's the same exact process, like very, very stringent list of questions. And we're very big on informed consent. A lot of the people are just very excited and like really want.
We do also have a whole regulatory team that's one of our questions ethically, is making sure that along the way, we're doing this as ethically as possible. So that gets a little bit on the line of proprietary stuff, so I'll tell you what I can. We're on a time scale of weeks, like in terms of growth and stuff like that. But like I mentioned, these cells aren't designed to live forever. They're designed to live, serve their purpose, apoptose, and die. So that's one of the challenges that in bioprocess I run into a lot. In terms of milk that comes out of this, and Shelley said it, but what we're making isn't at this time milk. It's a complex milk-like product, and we're trying to get as close to human breast milk as we can. But in terms of the volume and composition, we're still very much on the way there. Sorry, I can't answer in more detail. I love to nerd out about the science of it, but I think my boss might be a little mad at me.
Yeah, so I just wanted to ask, like, what, do you think? Like, where's
Yeah, that's honestly not necessarily what either of us
That's
do.
Not really hard to feel too much.
So, I would be happy with somebody on their team. But somebody on their team could start.
Yeah, I can think of a couple of people who can answer that.
It's gonna be so different. Yeah, no, definitely, there are people who think.
In our company, we just aren't them. You.
Yeah, I mean, I would say, like I said, I came from cancer genetics and then type 2 diabetes genetics. I knew nothing about lactation before joining, but, had a public health background, very, very interested. I got very frustrated with academia because I felt like it wasn't doing enough fast enough. So for me, it was just really important to find, a new job that was going to contribute more, and do more. And I think for, everybody there, it's a very selective process, as far as going through multiple stages of interviews. But I know, like, my first day, I was telling Logan yesterday, like, I just felt like I could found my place, and just fit right in. And it's definitely a culture that of just people who just want more change sooner now, like yesterday.
I've gotten that same question in a lot of interviews, like when they ask me, why do you work at BioMilk? And I've always felt very strongly about my answer. Coming out of my postdoc in grad school, I mean, I was always interested in cell agriculture. My background, again, material science, BME. I also like to cook barbecue a lot. So making food and science come together was something I was super interested in. But my real thought was coming out, moving out of academia and getting older, I realized that I'm probably never going to win a Nobel Prize. Like I made peace with that as I got older as a scientist. But when I was choosing between jobs, going to a place like BioMilk where I'm helping an underserved portion of the population, like Shelley's slide said, there's just not enough research done on this point blank. Like that's true. So if at the end of my career, if this is what I did, if I help this one portion of the population, that's good. That's a good use of my skills.
I was gonna ask that.
Sure.
Yeah.
Someone in the chat asked if you would be able to add in that type of probiotics.
So what we add in is, so that's going to be an evolving question as what the cells are making versus, and this is a question that formula companies have to answer as well. Getting a balance between costs, like what's the cost of putting the probiotics in and things like that to make your product accessible, but also have it as close to the nutritional profile that you need.
And pasteurization impacts it a lot too. Would you add it before pasteurization or after? If you pasteurize, it's going to kill everything.
So we're not sure yet, but it's definitely an avenue that we're thinking about.
Yeah.
And also for the Irish, I think you mentioned that the cells die immediately, so you guys work on it.
We're getting more, again, closer to the proprietary stuff. So it's something we think about a lot. And just to clarify, the cells, when they go into the bioreactors, they don't immediately die. There's a shelf life.
Like weeks and weeks and weeks.
Yeah, there's a shelf life where eventually enough apoptotic signals are being sent out that maintaining a viable culture gets very difficult.
I mean, general, people cell aggregates that out there from cell the I guess it's also here.
Yeah.
Yes.
I mean, is there a way to find?
Yeah, so spontaneous immortalization does occur. So that is, something, but it's also, you can consider, I think the average lifespan for cells that are coming from the milk, without any kind of intervention, it's weeks. So there's ways to do it without any kind of intervention as well.
I think I remember on the website, something about, it was kind of used for a thing, or something that might get individual cells and then sell on their own.
Yes.
So that it might actually have, these for that.
Right.
So specifically to that, it's something that, again, and it's going to be in the
Yeah.
future. Right now, we're kind of getting the bioprocess to the point where we can get scalable and actually have products. But it's something that we would love to do. Yeah, absolutely. A lot of extra questions get added in there, like how do you safely extract samples for someone who isn't lactating yet, because it's going to take time to get the cells in a state where they can produce biomilk. So it's something that will is a good deal down the road, but it's something that I personally am super interested in when we get there.
And another thing is to, again, make that. But it is certainly something that we think about. It's very interesting. There's a lot of future applications. Something that I think about a lot is if we could make it actually more shelf stable, more convenient, and have a lot of implications for famines and countries where there's malnutrition, civil unrest, those kinds of things, with getting a very, very necessary supply that could actually be better in every single way in terms of practicality. There's a lot of directions that we can take this in the future. Yeah.
Yeah, I was just wondering the advantage of the package personalized, because usually if the antibodies are made with lymph nodes, she wouldn't necessarily be part of it as well. They would be more, whatever antibodies were in the current sample would be stable.
Right, it was processed out of the liminal.
Yeah, I wonder if there are advantages of having something personal to you.
I mean, I don't know if you have thoughts about this, my honest answer is that, I don't know that it's necessarily from like a health perspective, that it's that much more beneficial, but I could see that being something where somebody wants to, you know, really struggle with breastfeeding, it would be probably more significant than I would imagine. Like I'm still giving milk to my baby, so my,
Yeah, that makes sense.
yeah.
And just for like the secretary profile, like we mentioned, we have 60 to 100 different lines, and they behave very differently. Like
Yeah.
it's a very like from donor to donor, it's extremely different. So who knows? Like we could get like one donor who's like just when we take it, it's amazingly easy to put it in the bioreactors, and with some cells they don't like it. Any other questions? All right, well, feel free to, we'll leave this up so you can take pictures. Feel free to come get a card for both of us. We both answer our emails fairly regularly. So I think the QR code for our personal LinkedIn's are in there as well.
Great. Y'all enjoyed this, I mean, if you weren't.
We will take one with us. Thank you online for everybody. Thank you.